Welcome back. This is the fourth section on the central control of reproduction, and this one's all about the pineal and seasonal breeding. And we're going to be looking at how light and dark controls what's going on, not only within the brain, but within the reproductive system as well. You should be familiar with this slide by now. Here we have the black box, which is essentially the brain, in which we have the GnRH neurons coupled together in some way as a GnRH pulse generator with an output which drives the reproductive system. We know there are many inputs into this system, and this time we're going to be looking at how seasonal factors can control reproduction. To understand seasonal breeding, we have to actually understand circadian systems as well. It's all to do with how timing controls reproduction. And we know in terms of the circadian system, there are many circadian systems in humans: the digestive tract, blood pressure, body temperature, sleep, hormone levels. Anything you look at, it's not stable. It varies throughout the 24-hour day. And these rhythms are normally coordinated together by some kind of mainly central clock mechanism, although that doesn't rule out individual systems having their own little clocks going on within them. And the central clock being driven essentially by the light-dark cycle coming in, acting as it's called a Zeitgeber, a time giver for the circadian clock. So what do we know about the circadian system? Well, we know that in humans and in most animals, it's driven by the light-dark cycle. Light comes into the eyes, but how does that light-dark cycle control and set the circadian system? Our understanding of this has developed remarkably over the last few years. And this is actually a cross-section of the back of the eye, back of the retina. So light is coming in from this way, it hits the retina, and the traditional light-sensitive cells, the rods and the cones, are down here, sending their nervous signals up this way to the retinal ganglion cells, which go via the optic nerve to the rest of the brain. But there's a subset of these retinal ganglion cells which seems to be absolutely essential for the circadian system in mammals. It's a subset of photosensitive retinal ganglion cells. Photosensitive rods and cones are photosensitive, those are the traditional ones, but some of these are photosensitive as well. Why? Because they've got a special light-sensitive pigment called melanopsin. And melanopsin is especially sensitive to blue light. And it's these cells which seem to be the primary time givers to the circadian system inside. They send their messages via the optic nerve to the hypothalamus and to the suprachiasmatic nucleus, which we'll see in a minute is the central clock mechanism. So here we have the whole system. Here we've got day and night. So the alternation between day and night sends the signals via the photosensitive retinal ganglion cells, and the optic nerve, within the optic nerve there's a special retinohypothalamic tract which leads straight to the hypothalamus and inputs into the suprachiasmatic nucleus. Now the suprachiasmatic nucleus within the hypothalamus is the site of the main circadian clock mechanism. Really, it's called the master clock. Every cell may have its own little clock. Every tissue may have a collection of clocks, but the suprachiasmatic nucleus is the major coordinating clock in the whole body. The role of the suprachiasmatic nucleus in controlling the circadian rhythms throughout the body is partly via hormonal mechanisms, controlling, for instance, the circadian output of the anterior pituitary. But a major role is also by controlling the circadian output of melatonin. Now the hormone melatonin comes from the pineal gland, and the suprachiasmatic nucleus controls the pineal gland via a pathway which goes from the suprachiasmatic nucleus through other nuclei within the hypothalamus, down the vertebral column to the superior cervical ganglion, and then up to the pineal gland. So the superior cervical ganglion is a major link in the pathway to the pineal gland. And here we see the general profile of melatonin in humans as you go through the day. This is midday here, 8 p.m. As darkness comes in, so melatonin levels go up, they peak and they come down again, and in the morning melatonin levels drop to normal. So a daily rhythm of melatonin production, really driven by the light-dark cycle. Let's look at the inputs then of the circadian system into the reproductive system specifically. One input is via the suprachiasmatic nucleus. As well as having many outputs, generally one of its outputs is essentially onto the GnRH neurons via the kisspeptin neurons which feed into it, and by neurotransmitters such as VIP. They control the output of GnRH and the circadian output of GnRH. But we're going to spend the rest of this lecture looking at how the pineal and melatonin control reproduction. The pineal gland used to be thought of 50, 60 years ago as really pretty useless, didn't really do much at all. Then, because of studies on circadian rhythms and because of studies on seasonal breeding, it began to be realised that the pineal gland played a very important role in normal physiology. What we're going to try and explain in the next few slides is this. In temperate regions where there's a marked seasonal variation between winter and summer in terms of day length, not just day length, but also in the temperature, the rainfall, often vegetation patterns, food availability, animals will tend to produce their young at the optimal time of year for their young to grow and survive into adults. And that's usually in the spring. So what we see is animals will give birth in the spring, or they'll hatch out their eggs in the spring, and then depending on how long their pregnancy is, either they'll mate at this time of year, or, for instance, sheep who have a long pregnancy, they mate in the autumn, so they give birth in the spring. Birds have a relatively short incubation period of 3, 4, 5 weeks or so, so they'll mate, egg lay, hatch out, so the young are available to survive and make use of the good conditions in the summer. So long day breeders breed when the days are lengthening to make use of the summer conditions. Things which are called short day breeders, such as sheep, they breed in the shortening day lengths of autumn to give rise to the offspring surviving or being delivered in the spring, so they can survive through the summer and make use of the summer conditions there. Day length is a very consistent seasonal signal, much more so than the variations you get in, for instance, rainfall or temperature or vegetation. Day length, the same year after year after year. And we're going to see that this is reflected in the daily melatonin levels. These are melatonin levels in the sheep, exposed to long days, short nights, and we can see during the day melatonin is very low and it goes up at night. But this is a short melatonin signal compared to this one, with some long melatonin signal. This one's got a long melatonin signal because it's got short days and long nights. So melatonin is coming out at night. So the consistent day length signal is reflected in a consistent melatonin signal. Here we're looking at the drive of day length in the reproductive control of sheep. We're going to be looking at the Soay sheep, which are an ancient breed of sheep, and we're looking at seasonal changes in their testis diameter. Now remember, these are going to breed, that is mate in the autumn, and we see that in the autumn, here we are, September, October, November, large testes with decreasing day length. Then as you go through into spring, then the testis goes down in size, and then as the day lengths increase again after the summer solstice, they reach their maximum again, and so you go cycle after cycle, with the testis size being driven by the daily changes in day length. We can really show that it changes the changes in day length by driving it artificially. This is done, top one is just the natural changes as you go through the year. Let's speed it up a bit. Put the sheep in artificial conditions and expose them to artificial day lengths. Here we are, short days, long days, short days, long days, short days, long days. And we can see we're driving the testis size much quicker than happen naturally. In short days, the shortening days, typical of what would be autumn, testis gets big. In the long days of summer, then the testis regresses. Here we're looking at the drive of day length in the reproductive control of sheep. We're going to be looking at the Soay sheep, which are an ancient breed of sheep, and we're looking at seasonal changes in their testis diameter. Now remember, these are going to breed, that is mate, in the autumn, and we see that in the autumn, here we are, September, October, November, large testes with decreasing day length. Then as you go through into spring, then the testis goes down in size, and then as the day lengths increase again after the summer solstice, they reach their maximum again, and so you go cycle after cycle, with the testis size being driven by the daily changes in day length. We can really show that it changes the changes in day length by driving it artificially. This is done, top one is just the natural changes as you go through the year. Let's speed it up a bit. Put the sheep in artificial conditions and expose them to artificial day lengths. Here we are, short days, long days, short days, long days, short days, long days. And we can see we're driving the testis size much quicker than happen naturally. In short days, the shortening days, typical of what would be autumn, testis gets big. In the long days of summer, then the testis regresses. We're now going to look at the evidence that the pineal is involved in these changes. So here are control rams under artificial photoperiods, short days, long days, short days, long days. Here we can see those photoperiods driving testicular diameter. And this is a system we're looking at. I know it's rather small, but the key point is the light dark information is coming in via the suprachiasmatic nucleus, goes down to the superior cervical ganglion, which innervates the pineal gland. So if we get rid of the superior cervical ganglion, we get rid of the innervation to the pineal gland. And this innervation is key to driving the melatonin secretion. So we're now going to get rid of the superior cervical ganglion. Here we are. We've taken it out. The pineal gland no longer has the innervation from it. And what's the response of the sheep? They no longer respond to the short days, long days. It's important to remember that melatonin is just a signal. How animals interpret it will depend on the species. In species with long pregnancies in mammals, the long pregnancies mean they breed in the autumn, and it's the lengthening melatonin signal in the autumn which turns on reproductive systems. In the rapidly breeding rodents, where pregnancy may be only 21 days or so, or even less, and a hamster is only 16 days, it's the long days of summer, or the lengthening days in the spring, with a shorter melatonin signal, which turns on reproduction. We're going to switch our attention now to the hamster, which does breed in the summer. So in the long days of summer, short melatonin signal, reproduction is turned on. In the winter, the longer melatonin signal turns reproduction off. And we're going to use this model to actually look at how we can prove that melatonin really is an important switch mechanism. We're going to do this by injecting melatonin into the animal, and depending on the time you give it, then we can change the duration of the signal. So this is the normal situation, a short melatonin signal in the spring, summer, and a long melatonin signal in the winter, and the winter signal turns reproduction off. So what we're going to do is inject melatonin. We're going to inject it in here to lengthen the short melatonin signal. Here we are, injecting it in. So we now have got a long melatonin signal, and what's the response? Reproduction is turned off, just as it is in the winter. So it's the duration of the signal and how the animal interprets it which determines whether reproduction is on or off. While our focus is on reproduction, it's important to remember that seasonal reproduction is not just about reproduction itself. It's about survival. And animals have to change their physiology as they go through the year. In preparing for the winter, animals may store huge amounts of fat. They change their behavior, they may change their diet, they change their metabolism, they can change their plumage or their fur. And this requires a lot of coordination within the body. So how is seasonal reproduction linked in with these other changes? Well, in trying to study this, something very interesting came up. And it was obvious that the thyroid was involved. Now that's sensible in terms of changing metabolism, etc. But it was shown that thyroidectomy of both birds and mammals seemed to block seasonal reproduction. So where is the thyroid involvement, or how are thyroid hormones involved? Well, it turns out that melatonin and thyroid hormones are closely linked. This came from studies of where melatonin acts. Now, melatonin is a circulating signal that goes all the way around the body, but one of its major binding sites is in the pars tuberalis. Now, the pars tuberalis is essentially the little bit of the pituitary stalk, but the pars tuberalis is the part of the anterior pituitary which wraps around the pituitary stalk. So here we have the hypothalamus leading down into the posterior pituitary. Here's the anterior pituitary, and this bit here wrapping round the pituitary stalk is the pars tuberalis, and it's a consistent binding site for melatonin in seasonal breeding mammals and birds. So what does melatonin do when it binds here? Well, the pars tuberalis has a high percentage of thyrotrophes, cells which secrete TSH, and if they secrete TSH, then the question is, what is the TSH doing? So this is a situation we now have. The light-dark cycle, detected by the photosensitive retinal ganglion cells, information to the suprachiasmatic nucleus, the master clock, one of whose outputs is via the sympathetic nervous system to the pineal gland, which produces its daily output of melatonin coming out at night. The melatonin acts in many places in the body, but also on the pars tuberalis, and affects its production of TSH. Now this TSH, rather than acting systemically via the thyroid, actually acts locally within the hypothalamus. It seems to get to the hypothalamus probably by a little bit of retrograde blood transport from the portal blood vessels, but also via tannocytes, which are specialized ependymal cells which have long projections down towards the median eminence. The role of TSH within the tannocyte seems to be to control the levels of T3 and reverse T3. T3 being the active thyroid hormone, reverse T3 inactive, T4 being derived from the thyroid gland itself. So what the TSH does within the tannocytes is control the activity of deiodinase 2 or deiodinase 3. So controlling the relative availability of T3 or reverse T3. And those two hormones in turn act within the hypothalamus to cause various changes, neurodegeneration, neurogenesis, remodeling. These have all been shown to be affected by the thyroid hormones in controlling all the kinds of things which you see in the seasonal cycles. And it's noteworthy that the TSH-driven changes in deiodinase 2 and deiodinase 3 is a strongly conserved photoperiodic mechanism in both birds and mammals which breed seasonally. Circadian rhythms and seasonal breeding are fascinating subjects and deserve much more attention than I've had time to give them. But it would also be wrong if I didn't just add this one, that mammals and birds actually are not identical in terms of how their mechanisms work. Here we have the case in mammals, where we have the light-dark cycle via the eye, via the suprachiasmatic nucleus, via the pineal, the melatonin signal, the melatonin acting at least partly on the pars tuberalis and TSH, deiodinase 2 and 3, affecting T3 levels within the hypothalamus and controlling many things there. That's mammals. Do birds work similarly? Well, in some ways they do. TSH works on the pars tuberalis, affecting deiodinase 2 and 3 in birds as well. But the control of that TSH isn't quite the same. Birds, the pineal may be far less important, and birds have relatively thin skulls, and there seem to be deep brain photoreceptors, so different from the photosensitive ganglion cells that we saw in mammals, but there are deep brain photoreceptors which may well mediate this light sensitive response of TSH within the pars tuberalis and then affecting T3 levels within the hypothalamus. So birds and mammals may have similar hypothalamic mechanisms, but how they detect the light, or the light-dark cycles, may be rather different.